Alexa, stop. Okay, just getting psyched for my lecture. Alright, so what we're going to be doing today in this lecture is looking at one of two things. The first lecture is going to be over epithelium and secretion. So what we're going to be addressing is glandular epithelium, one thing that we kind of skipped over as we moved into looking at connective tissue proper and the other types of connective tissue. So for this lecture, we're going to kind of go back and we're going to revisit epithelium, and we're going to talk about what it does for the body in terms of secreting different molecules for everything to function properly. So what you guys need to do in your notebooks, we're on pages 16 and 17 for the note set that we're going to be doing today. It's the page that's after our notes that we took on epithelial tissue and the properties of epithelium. If you did not leave a page blank for that, you just went on, that's fine. Just find your next two pages that are blank side by side, and you can go ahead and follow along with the notes there. So I'm going to go ahead and get the lecture notes up. So I'm going to show you guys our notebook that we're working on. Alright, so what we're going to be addressing today is epithelium and the secretions that are produced by that epithelium. So what we're going to start with is just talking about what essentially this epithelium is. Okay, so we've addressed the different functions of epithelium, so protection, aligning tissue, it's there for receptors and things like that, but another major function of epithelium is to produce for the body major molecules. So your epithelium is actually secreting quite a bit of what your body needs in order to function in a normal homeostatic state. So what you're going to see today as we address this, we're going to be looking at the different types of epithelium that line the body that secrete different stuff. And we'll talk about the different secretions and the different methods in which secretions occur. Okay? So a major function of epithelium is to produce many of our body's molecules in order for it to function properly. Okay? So again, we know that epithelium does these things. It helps with regulation because it controls what goes into and out of our bodies. Right? It's there for signaling, right? It's that first step when we're getting our signaling going on. So it's that first receptor molecule that we're going to have until we continue that on through the nervous system. But again, it's there for protection. Okay? So again, besides just excreting things, these secretions help with that regulation. They help with signaling, and they help the body protect itself. Alright, so let's define what a gland is. Okay. So a gland is any organ that secretes chemical substances. And so that could be anything as small as, say, a sweat gland or a sebaceous gland that's producing oil on the skin, to things like your pituitary gland or mammary gland or your ovaries and testes or your stomach or your pancreas. Those are all organs that secrete chemical substances. So they all fit within this category of being called a gland. So within inside of a gland, you're going to have different types, and that's just dependent upon where the material that is produced is going to end up. Okay, so how it actually gets transferred. And so there are two types of glands that exist inside of the body. Okay? The first type of gland that you're going to see with our substances is called an endocrine gland. So endocrine glands are extremely common in the body. Okay? And what sets endocrine glands apart from other glands is that with an endocrine gland, you do not have a duct in which you are secreting materials. You actually release these materials into the interstitial fluids around the cell. So remember, with epithelium, epithelium is going to release that into the interstitial fluid, and it's going to get passed through diffusion to various parts of the body. Okay? So with an endocrine gland, we're releasing hormones into that space. So your cells are going to react to that increase in the hormone, right? Pass it along through diffusion and carry it from place to place. Okay, so that's an endocrine gland. So there are no ducts that are present. Okay. The other type of gland that you can have that produces secretions is called an exocrine gland. So exocrine is outside. Exo means to the outside. And what these do is produce secretions onto the epithelial surface. Okay, so they actually secrete it to the very top of the cell, so to that apical side of our cell. And so what it's going to do is generally produce material into a duct. So you're secreting that substance to the top. That substance in the top is going to collect that fluid or material that's being produced, and it's going to carry it along in a duct. This duct can be something like as small as like a few layers of cells that are going to pass it on to the bloodstream to get that to occur very quickly, or it can secrete to something to the outside that's going to then get carried to the outside of the body, such as like what you're seeing with your sebaceous glands and things like that. Okay, so again, endocrine, no duct. Exocrine, there is a duct. Okay, and by duct, I'm referring to a epithelial line tube, right, that's going to have a lumen inside of it. So if we're looking at a slide of an exocrine gland, you're going to see that lumen in the middle. Okay, so we saw this, for example, when we looked at our stratified cuboidal epithelium, when we looked at parts of that secretion, okay, so, or that organ, all right, we were actually looking at a gland, right? So that was actually an area that had those two layers of cells for that epithelium, okay, that stratified epithelium that was cube shape, and then that hole in the middle with which the lumen was at, all right, so that apical side is pointing to the lumen, it's secreting that material directly into that gland. All right, so with secretions in our endocrine and exocrine glands, we actually have lots of different mechanisms. There are actually 3 different mechanisms that your body will use to secrete materials. Okay, so we'll go over those 3 mechanisms briefly, then we'll go through all the steps of them on our other page. All right, so our first mechanism for secretion is called Apocrine secretion. And so with all of these different types, we'll talk about how the material is produced, but essentially this is all about how that material gets passed on to the next step, right, to where it goes. So Apocrine secretions are secretions that are produced inside the cell, right, and they get packaged in the Golgi. Okay, so the Golgi apparatus, if you remember, is there to take materials and wrap it up in a membrane-bound sac called a vesicle. Right, and what happens in the Apocrine gland is that those vesicles exocytose. So they float to the top of the cell, to the apical side. Once they reach the apical side, then they're going to actually attach to the cell membrane and then release that material out. Okay, so just grows the cell membrane for a little bit, sends that material on its way, the cell lives, all right, and it can rapidly do this. So your sweat gland is an example of a Apocrine gland. So it's actually releasing this fluid through fluid as well as other different things to the surface of the cell out so that the body can use it to help cool itself off. Okay, that's Apocrine. So remember, Apocrine is vesicles. Okay, Apocrine is vesicles. Our second mechanisms that we're going to have for secretion is an apocrine secretion. So apocrine, A-P-O-C-R-I-N-E. Apocrine secretions are again where you have your molecule that's produced in the Golgi, or produced by the cell. It's packaged up in the Golgi, right? And then those Golgi are producing vesicles. And what's different from apocrine compared to Apocrine is that in an apocrine cell, you're actually producing a shedding of the cytoplasm. So you're actually cutting off part of the cell in order for that material to be released. So you actually use cytokinesis to bring in the sides of the cell, pinch that part off that has all those vesicles in it to then be released to the outside. So part of the cell does get removed and essentially dies away, but the remainder of the cell is still alive. So with an apocrine secretion, part of the cell is still alive, right, and is allowed to grow again, to reproduce more of these different vesicles full of the material you want to excrete and send it on its way. So with an apocrine gland, an example of this is the mammary gland. So men and women both have mammary glands, but the mammary glands are the ones that produce milk in mammals. Okay, that's where we get our name from. So we produce milk because we're mammary. So mammary glands are ones that release an apocrine secretion. So you end up building up the milk molecules in these vesicles at the top of the cell. That top of the cell gets sloughed off by cytokinesis, bursts, allows that material to then release, and the cell can keep growing and producing material. Yeah. All right, so our last type of cell is one that's kind of sad. All right, but this is happening when your cells lyse. Okay, so a holocrine. Holocrine, H-O-L-O-C-R-I-N-E. Holocrine glands are secretions that are released by the cell through lysis. So remember, if a cell lyses, that cell ends up dying. And so these are cells that die for the cause. They will die, they will take the whole of it, and they will die and kill themselves to produce that material. So once they build up their molecules inside of the cell, that cell will just burst open and release everything that's contained within it. So this ends up killing the cell, right? But the cells are replaced by stem cells, and these stem cells are going to be found on the basal lamina of our epithelial layer, so at the very base right above our connective tissue. So these holocrine secretions are going to be released directly out of our cell because the cell is just going to burst open. So sebaceous glands are a really good example of a holocrine secretion. So to produce that oil that's contained on your skin, or what you use, or what your body produces to keep your hair supple and moist, if you don't wash it out 24/7, is that your sebaceous gland is generating all of this oil, and then it ends up bursting, releasing all of that oil very quickly to allow it to then be excreted through a duct. Okay. So again, with holocrine secretions, the cell dies. Right? So Apocrine is vesicles, apocrine is shedding, holocrine is you are wholly dead. Okay. So holocrine glands kill the cell. Alright, so that's basically it when it comes to our different types of epithelium and secretions. So I do want to go over the steps that are involved in this. So one of your essay questions on the test will be to describe the mechanisms and how one of these three types of secretion occurs. Okay, so if you take your notes and flip to page 16, so the page next to it, right, I do have a couple foldables on this page. So you guys got a sheet already that has this image on it. If you do not have it, there are extra copies at the front of the room that you can grab so you can have it to fill it out. Okay, so this is just a supplement to help kind of diagram what it looks like for each of these different steps. So I just went ahead and glued it to the edge here so that I can flip it over and get to my notes on the steps of different secretions. You also today got a table on the classification of exocrine glands. So again, a table that's summarizing the basics of what I'm talking about here, okay, but in a different form, so you can use that to help you study very quickly. Okay, but I do want you to write everything down on your notes because I think it'll be beneficial for you to study. Okay, so let's go back up and talk about the steps of secretion. So in order for something to be secreted, right, it's going to carry out one of these three steps. Okay, so if you have Apocrine secretion, that's different from an Apocrine, that's different than a Holocrine. Okay, and they all carry different steps. So for Apocrine, the three main things that happen are your cell first produces the secretion. Okay, so your cell is making whatever molecule you need in order for you to produce. In this case, say we're preparing for sweat. Okay, that sweat gets packaged by the Golgi and forms these vesicles. Right, again, the vesicles, as they're ready to be released, will travel to the membrane. The membrane gets a signal to allow for excretion because the vesicle will attach to the cell membrane. It will open up and release the material into wherever it's supposed to go, so into a duct or whatever it's supposed to go to. Okay. All right, so that's the basics of Apocrine. So very fast, very quick. Okay, so production of secretion, you package that material in the Golgi and the vesicle, and you travel to the membrane, and in the membrane you are releasing that particular material. Alright, so let's go to our second type. Alright, so apocrine. Remember, apocrine is where our cells are shedding part of the material. Okay, so you're not completely removing, or you're not completely killing the cell, you're only taking off part of it. The cell can regrow and continue to produce materials necessary for secretion. So in order to have apocrine secretion, the first step that happens is your molecules are being produced by your cells. Once those molecules are produced, they're again packaged in a Golgi, or packaged by the Golgi apparatus and sent to the vesicle. You get an accumulation in the apical side of the cell, so you're allowing more of that cell to be produced, all right, producing that material, and then cytokinesis will occur. Cytokinesis at the top of the cell is going to essentially chunk off that top part of the cell and then release it out into wherever it's going to go, so into a duct or onto the surface. So again, with apocrine, if we're talking about in the mammary tissue, it's going to go to a duct. That duct is then going to go to the nipple and release the milk. Okay, so cytokinesis is going to occur at the top, release those vesicles into the secretion, and then that material can transfer. So remember, with apocrine glands, your cells are not dying, only part of them is being removed. All right, so then our last example after apocrine is holocrine. So holocrine, remember, this is where our cell wholly dies. All right, so we have one production of secretion. Okay, so your cell is producing the material yet again. Okay, but again, we're not packaging it in a vesicle. The cell grows in size because the cytoplasm is constantly collecting the material here. So then cytoplasm is released by the cell, right? Or because the cell is lysing open, right? So your membrane is actually bursting open and your cell is releasing that material into your duct or onto the surface of the cell. Right, so those stem cells, again, at the bottom will continue to be replacing these cells that keep dying. So your stem cells divide, replace that material in the cell, and they will continue to do so until that cell gets replaced. Once that cell gets replaced, it grows, releases material, and boom, it dies. Okay, so it's just the cycle of holocrine glands. Okay. So inside each of these glands, there are different types of secretions and materials that can occur, and that's all based on essentially the viscosity of the material, how dense or how loose the material is. So I do want to talk about the three types of secretions that you can have inside of a cell. So the first type of secretion that you're going to have are those that produce kind of watery secretions or thin secretions. We call those types of glands serous glands. Serous glands, generally a serous fluid is something that's very loose, very thin. So think like sweat. Sweat is an example of a serous fluid. It's very thin, it carries a lot of dissolved substances, and it can move very quickly. The second type is called a mucus gland. A mucus gland is going to secrete what we call mucins, essentially that stuff that makes you all stopped up. Right? So mucus glands are producing this kind of very dense material. So you have mucus glands that line your respiratory system, right? So that's like why you snot up. But you have mucus glands that line a whole lot of your different body systems. Anywhere where there's essentially needs to be lubricant for your body to function, or if it needs a dense layer for protection. So like the inside lining of your digestive system is all made up of this mucus gland. Right, so you're producing this thick mucus around your digestive system so you don't end up damaging the cells that are making up your stomach. And then you have glands that are capable of both. So we call them mixed exocrine glands. So they produce both a serous fluid and a mucus fluid. So examples like this are some of your skin secretions. They're kind of mixed. Your stomach is a good example of this serous and mucus secretion. So it produces serous type of fluids that are full of HCl that allows for chemical digestion, but also very much producing this mucus layer to help protect your stomach from more dangerous digestion. So let's go back to our image, our foldable that we have here. So I just went through and labeled the 3 different types, all right, and it kind of lists the steps and shows you a diagram of what's happening. So our Apocrine secretions, again, are releasing from the Golgi. Right? Our vesicles go to our cell membrane, those get released out, and our material gets transferred. With Apocrine, remember, our cell is producing the material, and vesicles, again, those collect at the top. Right? Cytokinesis occurs here, right at the very beginning. That part of our cell gets removed, and our cell grows or replaces itself and just repeats the cycle. And then lastly, remember, with Holocrine secretions, with Holocrine, our cell dies. So we have these stem cells at the bottom. The stem cells will continue to divide. That's step 1. Right? As those cells divide, they will build up material inside of the cell cytoplasm or in vesicles. That can also happen, but mostly in the cytoplasm. And then once that cell reaches the top, your body needs that material. That cell will get a signal to lyse, so it bursts open and releases all the materials it has into wherever it's going to go, the duct or the bloodstream or onto the surface of the cell. All right, so that is it for talking about our secretions. Okay? So if you have questions about this, right, in order to, whatever you need to help look at this, please feel free to ask. Okay, so this is what essentially I'm here for, right? So if you have specific questions about this, please let me know. If you need to look at stuff again or review stuff, just go back. I know I talked really fast, but again, just pause it, write down the stuff that you need. Again, if you have any questions, please let me know. All right, so good luck.